Welcome to today's lesson on producing monoclonal antibodies. In this session, we will uncover the specialized process that underlies how these unique antibodies are developed for medical and scientific purposes. In our lesson today, we will specifically focus on describing how monoclonal antibodies are produced. We will break down this step-by-step method, including the role of hybridoma cells in monoclonal antibody production. So far, you have learned about antibodies and their role in the specific immune response. You know that antibodies are critical components of our immune system, helping to identify and neutralize pathogens, such as bacteria and viruses. This knowledge about antibodies allows us to venture deeper into how scientists can harness and replicate these natural defenders in the form of monoclonal antibodies. Understanding this process not only broadens our grasp of antibody function, but also reveals how we can apply this knowledge in practical ways, such as in the development of targeted therapies for diseases. To start, let's recap the basics about antibodies. Antibodies are proteins that are produced by a type of white blood cell, known as a lymphocyte. Lymphocytes are highly specific, meaning that each antibody targets a unique antigen found on the surfaces of cells. This specificity is what makes antibodies so useful for targeting disease. Monoclonal antibodies are a special type of antibody that are produced from identical lymphocytes. These lymphocytes are clones of a single parent cell, thus ensuring that all of the antibodies that they produce target the exact same antigen. This uniformity makes monoclonal antibodies incredibly useful for a wide range of applications, including medical diagnostics and treatments. Producing monoclonal antibodies involves a unique challenge. While lymphocytes can create specific antibodies, they do not divide easily. On the other hand, tumour cells divide very rapidly. This makes them very easy to grow in large quantities. The solution to this problem involves combining the best features of both cell types. To produce the initial lymphocytes that generate the required antibodies, mouse lymphocytes must first be stimulated. To achieve this, scientists inject a small amount of antigen, the substance that triggers an immune response, into the mouse's body. The mouse's immune system recognizes the antigen as something foreign and responds by producing lymphocytes that make antibodies that are specifically designed to target and fight off that particular antigen. These antibodies can later be collected and used for various scientific or medical purposes. To produce monoclonal antibodies, the first step is to fuse these lymphocytes with our tumour cells. This fusion creates a new type of cell, called a hybridoma. Hybridomas have the best of both worlds, the ability to produce specific antibodies from the lymphocyte, and the capacity to divide rapidly from the tumour cell. Imagine a factory that makes a highly specialised product, but can't expand its production. By combining it with another factory that can easily increase production but lacks the specialisation, you get a new and improved factory that is capable of both specialisation as well as a high output. Once hybridomas are formed, they are cloned to produce a large number of identical cells. Since these hybridoma cells all originate from the same parent cell, they all produce the same monoclonal antibodies. The final stage of monoclonal antibody production involves collecting the monoclonal antibodies that have been produced by the hybridoma cells and then purifying them. This purified product can then be used for various applications, such as for targeting specific cells or chemicals in the body. Monoclonal antibodies can be tailored to target almost anything. One common use of monoclonal antibodies is in medical diagnostics. They are used in pregnancy and blood tests in order to identify specific biomarkers. A second use of monoclonal antibodies is in treating diseases. This is because monoclonal antibodies can target cancer cells, thus preventing transplant rejection and facilitating the treatment of autoimmune diseases. Finally, monoclonal antibodies are also essential for research. They are used to understand both biological functions and disease mechanisms. Before we finish, let's now go through a quick recap. Producing monoclonal antibodies involves the following process. Step 1 is to stimulate mouse lymphocytes with antigens to produce a specific antibody. Step 2 is to fuse these lymphocytes with tumour cells to create hybridomas. Step 3 is to clone the hybridomas to produce a large, identical population of cells. Step 4 is to collect and purify the monoclonal antibodies that are produced by these cells. In today's lesson, we covered the production of monoclonal antibodies, which are essential tools in medical and scientific research. We started by recapping the basics of antibodies, proteins that the immune system uses to neutralize foreign objects, such as bacteria and viruses. Monoclonal antibodies are identical copies of a single antibody, where each batch targets a specific antigen. These monoclonal antibodies are produced through the stimulation of mouse lymphocytes to produce specific antibodies. Then, the fusion of antibody-producing lymphocytes with tumour cells to create specific cells called hybridomas. These hybridomas are then cloned to form a very large number of cells, which all produce the same antibody. The antibodies that are collected from these cells can then be purified for use. Finally, we discussed the applications of monoclonal antibodies in diagnostics, disease treatment, and research. This highlights their significant impact on advancing medical science